right, I think we'll get started now. Welcome to Perennia's first ever virtual field days. We would definitely have rather been there in person and been able to see all your lovely faces in the field. However, we're glad to be able to still bring you some great information. So I think we'll get started. A little bit of housekeeping here before we get started. Everybody, all the participants have their audio and video turned off during the webinar. If you'd like to ask a question, please use the Q&A feature at the bottom of your screen, and you can put the question right in there. If somebody's already written that question, you can upvote it, like you can like it, and that will make sure that it gets to the top of the question thing. You can use the chat, but the Q&A is just as easy. And I will be monitoring that for our Q&A session at the end. Also, we are going to show some videos, and just as a heads up, the videos might lag a little bit at the beginning, but they should smooth themselves out, so don't panic. They should all work. We are offering pesticide points for today's session. So that's one pesticide point in pest management. You should have registered your pesticide applicator's license when you registered for this event, and you would have received an email. And keep that to submit with your records for renewal. So today, the topics that we're going to cover are Grape Phylloxera, squash bug and cucumber beetle, and then screening BT trials for corn and resistance development of corn insect pests. So on the line today, we have a great panel of people. So we have Francisco Diez, who is our viticulture specialist. Myself, I'm the acting Congdon. I'm the acting vegetable specialist. We have Sonny Murray, who is our field crop specialist. And then we have some excellent guests with us as well. So we have Dr. Deb Moreau with AAFC, Dr. Suzie Blatt with AAFC, and Dr. Jocelyn Smith with the University of Guelph. And in case you missed them, later this week, tomorrow, we will be talking about production systems, and Thursday, we're going to be talking about weed management. We have some other specialists and some other excellent guests, so make sure you tune in and don't miss those. So let's get started.
Good afternoon. My name is Francisco Diez. I am the Viticulture Specialist at Perennia. Today, we are here in the vineyard to talk about a small insect called Phylloxera. Here, I will show you some general characteristics of the insect affecting the vines, and also tell you a little bit about the history, the biology, and describing or differentiate it with other insect damage. After this video, we'll continue with some discussion with Dr. Deb Moreau, entomologist of AAFC, to know more details about this aphid, plus knowing some management techniques, how to handle it. Now we are located in other area of the property. This is Chardonnay, grafted in a rootstock of 3309. To continue talking about the aphid, Phylloxera, this insect is native of North America, and it was accidentally introduced to Europe in the 1860s in Europe, and the introduction changed completely the European way to cultivate and work in the vineyard. So different techniques, they have to be developed to mitigate the effects of the Phylloxera. And one of the biggest improvements or developments was the use of rootstocks. These rootstocks were used mainly to mitigate and to avoid to having more damage due to Phylloxera. Currently, nowadays, these rootstocks can be used not only for Phylloxera, also for manage drought situations, salinity situations, or acidity. So rootstocks can be used much more for other things. But right now, the main topic here is for the use of phylloxera. As I have mentioned, the insect was introduced accidentally in the 1860s to Europe. And one of the first countries to have more damage was France. So in 1878, France did a survey and detected that 1.2 millions of hectares or 3 millions of acres were affected by the aphid. So all that area had to pull out. At that time, all this surface represented the 50% of the acreage of France. After France and declared the aphid there, appears Portugal, Germany, Italy, and so on. So the insect spread quite fast throughout Europe, affecting the whole industry in total. How was introduced the insect? It's clear right now, the nurseries and the viticulturists, they try to manage other disease imported from the Americas and trying to find the management, bring it the insect. This disease was known in France as oidium. For us, it's powdery mildew. The proper identification of the insect and to know what was affecting the vines in Europe was only thankful to the collaboration of different professionals coming from different countries to know what was happening and what was the common patterns. Phylloxera, it's an insect, part of the group of Homoptera, and it has two biological cycles. One happening on the canopy, which can provoke galls, and the other under the soil, feeding from the root system and making some tumors at root system level. The adult aphid can be very small, from 1 to 1.25 millimeters of length, and their diet is specific to plants of the Vitis gender, but some species have developed some resistance. Usually, the American Vitis have more resistance to this insect. According to an scale, the most resistant Vitis varieties are Vitis rotundifolia, and the less ones are Vitis vinifera. Biologically speaking, the cycles of Phylloxera can affect differently if you have a Vitis coming from America or a Vitis vinifera. In the case of the Vitis coming from America, they have all the cycles on the canopy and on the root system. Meanwhile, the Vitis vinifera affects mainly at the root system. They don't have too much damage at the canopy level. In the canopy, where are the galls, inside of each one exists a larva. On the other hand, in Vitis vinifera, it's more difficult to establish at the leaf level, and mainly the whole cycle will happen only at the root system. So, which will be the results? The plants of Vitis vinifera, such as Pinot Noir, Chardonnay, Riesling, without rootstock, they can have low vigor, chlorosis, dry leaves. And at the root level, you will see some small tumors. Currently, we are in a vineyard with the main variety right now here as Frontenac Blanc, on root, without rootstock. And if you can pay attention to the canopy, you will see some galls. On the top of the canopy, you will see the most characteristic symptoms, the galls coming inside of the leaves, and as I previously mentioned to you, here inside you will see how appears or they are the larvae. It's very important to remind and have clear the difference with Iridium mite. In the case of Iridium mites, when they are affecting the canopy, the galls will not be inside, they will be outside of the leaf, and behind you will see like a slightly whitish condition. Again, as I already mentioned, in the galls of Phylloxera exists a larvae, which will continue in the canopy, but also they can migrate to the root system and continue their multiplication. Please continue in the session, because now we'll be discussing with Dr. Deb Moreau with more specifics.
Hi, I'm Caitlin Congdon, Acting Vegetable Specialist at Perennia. Squash bug and cucumber beetle are major pests of cucurbit crops, not only due to their feeding damage, but also because they vector diseases that can be detrimental to the crop. I'm going to highlight some information from a trial that is on the go this summer, looking at squash bug and cucumber beetle and some potential management practices to hopefully get the most out of your crop. But before we get into that, I'll provide a little background on each of the insects we will be looking at today. Cucumber beetles are the primary vector for bacterial wilt in cucurbits. Adults feed on weeds, grasses, and cucurbits before breeding and laying their eggs. In Nova Scotia, we typically only see one generation of striped cucumber beetle, although they tend to move into the field in flushes, so it can be problematic at multiple points during the season. Due to the high temperatures we have experienced in 2020, we have seen two generations of striped cucumber beetle, which will likely continue in subsequent years as we see more extreme heat in the region. Early season feeding damage can set the plants back before they even get a chance to take off, while mid-season feeding on the flowers of the plant can lead to blossom abortion and fruit loss. Bacterial wilt first shows up as dark green patches on the leaves. As the plant's vascular tissue is blocked with bacterium, the plant becomes wilted and eventually turns yellow and dies. Squash bugs have piercing, sucking mouth parts, which make them particularly adept at feeding on both foliage and fruit of the plant. Feeding symptoms on the leaf include yellow and brown spots, eventually causing the entire leaf to turn black and dry out under heavy feeding. Feeding can also lead to xylem vessels being severed, causing the entire plant to wilt. Feeding damage on the fruit can cause sunken, desiccated areas or scars, rendering it unmarketable. Squash bugs can also vector bacterial diseases such as angular leaf spot. There are 5 nymphal instars of the squash bug, ranging in size from 2 millimeters to 1.2 centimeters long. Adults continue to lay eggs throughout the season, especially in hot and dry conditions, so it is common to see several stages in the field at once. At least 3 of them can be seen here. Originally, we had hoped to conduct this trial on farm, but due to COVID restrictions, had to postpone that for another year. Luckily, Dr. Suzie Blatt with the AAFC, who is joining me for some discussion later, was able to put the trial in at the Kemptville Research Station so that we could get an idea of how everything was going to play out and what might need to be tweaked for the future on-farm demo. The trial is based on the idea of using a push and pull effect on the insects, either drawing them into a trap crop with the intention that they would therefore not do as much damage to the marketable crop, or deterring them with a companion crop that repels the insects, which in this case was nasturtiums. Nasturtiums have long been favored by gardeners for companion planting due to their tendency to repel certain insects and trap or lure others. This is great when you have a handful of cucurbit plants, but does it hold up over a more commercial acreage? The treatments that are being looked at in this trial are squash planted with a trap crop. The trap crop used was blue Hubbard squash, which is highly susceptible to squash bug. Squash planted with nasturtiums, squash planted with both nasturtiums and trap crop, and squash planted on its own with no nasturtiums or trap crop. This was repeated on both buttercup and butternut squash with 4 replicates of each. Here you can see nasturtiums planted alongside buttercup squash. Although there is some noticeable feeding damage, it hasn't killed the plant. There is some scarring on the fruit, which would render it unmarketable. You can see here what's left of the blue Hubbard trap crop is being thoroughly enjoyed by squash bug. Here's a clip from mid-August illustrating the damage to the trap crop.
So this is the trap crop that's been totally decimated by cucumber beetle. Now the good news is that any squash bug eggs that would have been on these plants likely didn't make it either.
This plot shows buttercup squash on its own, with no trap crop or nasturtiums. The foliage is wilted from cucumber beetle damage. Both squash bug and cucumber beetle are found to prefer buttercup to butternut, which can be seen clearly here. On the left is a plot of butternut squash on its own, with no trap crop or nasturtiums, compared to the same treatment but with buttercup squash on the right. Although the squash bug seems to have set up camp on this butternut squash, the fruit itself appears to be relatively unscathed. In fact, the only major fruit damage seen on the butternut squash is probably due to rodents or birds. Here you can see the range of damage to buttercup squash in this trial. This may be the result of toxic cucumber beetle saliva and clearly has the potential to destroy the marketability of the fruit, reinforcing the need to manage insect pest populations throughout the season. Thanks for watching!
Hi, I'm Congdon. I'm the acting vegetable specialist at Perennia.
I'm Sonny Murray, field crop specialist at Perennia. Today we're at Nick and Tim Lamb's farm in Berwick, quite a large dairy farm. They grow a lot of corn for silage, high moisture and grain. They do a little bit of alfalfa and a whole lot of grass forage, and then they also throw some winter wheat and soybeans in there. Today we're on the site of our corn plots. We have three trials going on this site. Behind us here we have the regional corn performance trial. It tests 34 silage hybrids, 10 long season silage hybrids, and about 32 grain corn hybrids. They're tested mainly on performance of yield and also dry down or maturity. Behind us here we have an AAFC trial looking at corn nitrogen rates and timing and their effect on corn test weight and yield. But what we're going to talk about today is the third plot we have in here, and it's what we call a sentinel plot. It has all the different available Bt proteins expressed in hybrids separately, so we can evaluate those proteins for the control of different corn insects.
So Bt, or Bacillus thuringiensis, is a bacterium that is found naturally in the soil all over the world. The Bt genes started being used in corn, put into corn varieties, and was commercially available from about the mid-90s on. Bt corn varieties were widely adopted in Nova Scotia. We have pretty high pest pressures here, so it was a good option for integrated pest management for farmers. So in 2018 in Nova Scotia, we found European corn borer populations that were resistant to one of the Bt genes or proteins, which has been a real challenge and a real game changer in the corn industry.
So there are 4 Bt proteins that are available in corn hybrids today. They're usually packaged together, so you're not buying one Bt protein in your bag of corn. You're buying at least 2 together. So why they package them together is it gives you a better range of control over the spectrum of insects, but it's also a resistance management tool. because if you have two Bt proteins in there that are controlling the same insect, it's very unlikely that that insect will become resistant to both of those Bt proteins at the same time. So as a farmer, it's very important for you to know what pests you have on the farm, what pests you're having trouble with, and what Bt proteins that you're purchasing from your corn seed. So it's very important that the farmer understands what insect problems they have on their farm, what they have for insect pressure, and then work with your corn seed dealer, retailer to know what's available to them so they can match the control measures to what insect pressures they have. And that Bt table is there to help you with that. Today in the plots, what we have is each hybrid is expressing the Bt gene in a singular form so that we can evaluate that Bt gene for the control of each one of the species of corn insects. It's very important to know if we're getting resistance to any one of those genes, because often on farm they're bundled together. So even though one Bt gene has full-blown resistance, it may be controlled by another Bt gene, which is very good for the farmer, but resistance can then happen very quickly because you only have one Bt gene that's giving you control instead of the two. You no longer have that resistance management built into that corn hybrid. So it's very important that we do this work to see if resistance is developing before you see it on the farm. So what we have here is northern corn rootworm. It's the Kawasaki green type beetle. We also have yellow variety around. That's the western corn rootworm. What this beetle does is it will come into your field at silking and start feeding on the silks. If this happens fairly early in the year before pollination occurs and the populations are quite large, it'll feed on the silks and you won't get the pollination of your kernels. You'll have bare spots on your cob. The real problem happens the following year. In Nova Scotia we have a lot of corn on corn acres. This insect will eventually go to ground, lay its eggs, and those larvae will emerge next spring and feed on the roots of the emerging corn crop. That'll cause poor performance and lodging in next year's corn crop. So in a lot of areas of North America, they don't do corn on corn anymore just because of that insect. So I just wanted to take a minute and review the different insects, what they look like, how to identify them, that we're looking at in the center plots. The first insect, and one of the most important ones, is European corn borer. It's been in North America for many years and a common pest of corn until we got the Bt gene back in the mid-90s. If you're seeing damage in your field, often the first thing you'll see is the tassel flopping over. And that's what we have here. You have your brown tassel, and where the European corn borer was feeding on the stalk, it had snapped over. Often you'll see it from the truck just driving by the field. You could also see tassels at the top of the plant breaking off and hanging down as well. When you're in the field, on closer inspection, you'll often see a little bit of sawdust down in the axis of the leaves. Collecting there, not to be confused with pollen that's coming off the top. This is a frass or sawdust that's been created by the borer as it's gone into the stalk, and you can see his little hole. And if we split this stalk open, we'll be able to see him inside there. Caitlin has a European corn borer there on the leaf, very recognizable by his dark, almost black head. Usually his body is a little bit whiter than this, but as he gets older, he will get a little bit darker. So they will overwinter in the field inside the stalk. One of the ways to control them is to shred that stalk, open it up in the fall, so that they'll perish over winter. What I wanted to go over is the western bean cutworm. One of the first things you'll recognize when looking for western bean cutworm is the shot holes, usually in the side of the corn cob husk. usually about the size of a .22 shot in the side of the husk. If you peel this off, we'll be able to find that insect. What happens with the western bean cutworm is the adult insects either emerge as moss from the soil, but we also have a population that migrates in. And we know this because in our trap catches, we'll see some very early trap catches. We think that is our local population emerging from the soil. And then midsummer, usually about the time that corn is tasseling, we'll have very large catches of a migratory population coming up from down south. So how to identify the insect? You find his shot hole, and then you start looking around, and you'll see where he's making a mess of the corn cob itself. You can see his feeding, and he's feeding down near the shank on this one, and you can see him there. So he's very easy to identify by the two black bands right behind his head cap. And usually you'll find, or not usually, but often you'll find more than one on the same stalk, because the moth, she'll lay her eggs as a cluster on the top of the plant. So the adult moth will be attracted to the tassel. She'll lay her eggs at the top of the plant, and then the young larvae will migrate down to the husk, where they'll burrow in and start feeding. So often, because they're laid as a cluster, you'll find many of them on the same cob or on surrounding plants. They are able to move from plant to plant, but they'll stay very local. The next insect is corn earworm. Corn earworm, it doesn't survive or overwinter in Nova Scotia, so it's a very migratory population, and it often is associated with jet streams coming up the eastern seaboard, or hurricanes will help push those moths up. When those moths get here, and we find trap catches starting very early in June, and they'll last right through to September. So last year we had a hurricane come through, I think it was the 14th of September or so. A couple weeks later, we saw huge corn earworm populations. Very late in season, they don't do a lot of damage when they come in that late, because they'll often just feed on the top of the corn, and the temperatures are very cool, so they're not very active. But when it happens quite early in the season, you can get quite a bit of damage. And they are quite large worms, and they come in a multitude of colors. The ones I have here today are green, but they can range from green to gray to more of a beige color. If you look closely at them, they're quite hairy and warty along the sides of the body, and again, they lack the two bands on the back of their head cap, the way the western bean cutworm does. So you can see they can get to quite a size. Now these guys, when the moth is flitting around laying her eggs, she lays the eggs on the corn husk individually. So these guys are solitary guys, very territorial, and they're cannibalistic. So you'll often find these guys alone on the corn husk. And if you put them together, at the end of the day, there'll be one. They are that cannibalistic. So, and they can do quite a bit of damage, as you can see. So there's another guy. Again, most of the guys I have today are all that green color. So you don't have much of an opportunity to spray these guys. The adult moth lays her eggs. 3 to 4 days, those eggs hatch, and then automatically the young larvae are down into that husk. And once they get inside the husk, you can't reach them with the spray. So very hard to spray, very hard to scout for, because you're looking for a single egg, so you're just not going to find them. So if these guys, again, arrive early in the season, they can do quite a bit of damage, a little bit of yield loss, but also just the quality of the grain. You got to increase the dust from their frass, but then you also have molds moving in, and some of those molds can be toxic to livestock and create feeding issues. So that is why we're really concerned about these, is the feed quality. So I think that wraps up the insect test that we were looking for. We saw a little bit of northern corn rootworm in the field. We saw European corn borer, western bean cutworm, and corn earworm.
All right, that concludes our video portion of the field day. If you have any questions for any of our specialists or guests, just remember to put them in the Q&A box at the bottom of the screen. And right now, I'm going to invite Dr. Jocelyn Smith to share her screen and tell us a little bit more about these corn pests and resistance issues.
Okay, can you see my screen there, Caitlin?
Yes, I can.
Okay, does that look all right?
Looks great, good to go.
Oh, great. Yeah, so I'm going to give you just a quick update on the different issues we're having with BT corn resistance and our major corn pests. Unfortunately, there's quite a few of them to talk about, but I'll try and keep it brief. So I just like to show this map of our corn production in Eastern Canada, the kind of a heat map of where it occurs. I'm down here in southwestern Ontario, south of London. And, you know, I think all of us in Canada are well aware that we've inherited most of these pests from the south over the years. And we're also starting to inherit a lot of the resistance issues that are happening where there's way more corn production further south of us. So Sonny talked about the different BT proteins in his video, and I'm trying to simplify this as much as possible for growers because I know it can be confusing. But there are really just four different proteins, as Sonny mentioned. The oldest one is Cry1AB. That was originally developed by Monsanto in the mid-90s to target European corn borer. And that Cry1AB protein is in a lot of the older hybrids. That was the trade names associated with it could be Yield Guard or Attribute. Probably around the early 2000s, I guess Dow at the time developed a second protein called Cry1F, and that's the one that is commonly known as Herculex in the trade, as its trade name, in Pioneer and Dow hybrids nowadays. Thirdly, this pyramid of Cry1A.105 and Cry2AB2 was released. And those are the ones that are found in the double stack hybrids originally, and then smart stacks and performance series hybrids later on. So these are really common. And when they're in these smart stacks hybrids and performance series, they are pyramided also with Cry1F. Okay. And the newest trait out there is the VIP3A protein, which Syngenta developed and is commonly known as Viptera or Leptra in NK and Pioneer products now. So just to look at the first, one of the three pests that Sonny was talking about was western bean cutworm. So I'm going to talk about this one first, just because it's kind of a newer pest to all of us. It came into Ontario in 2008, and it's been slowly moving your way for the last few years. It's been an issue in the East Coast. It's native to North America, however, and it's kind of been doing this eastward range expansion for the last 50 years. Now, we know that today the only BT trait that is effective on western bean cutworm is the VIP3A trait, and all of these others are not effective. Cry1AB was never thought to be effective, nor was Cry1A.105 and 2AB2. But in the early range expansion of western bean, Cry1F was seen to have some control. We in Ontario we're doing some trials looking at this in 2011 and so on, when western bean became a real issue for us. And you can see in some of our plots in 2011, we had about 90% of the plants in our plots with western bean cutworm damage if they were non-BT. But in the smart stacks plots that contained the Cry1F hybrid, as well as the Cry1A.105 and 2AB2 proteins, we had maybe 50% incidence. So there was significantly less damage, but it was never really great control of western bean cutworm. But the neat thing, well, not the neat thing, the unfortunate thing to notice is how quickly we went from having a little bit of control to no control. So by 2012, there was no difference in injury to Smart Stacks plants compared to a non-BT plant in Ontario. And that has since been confirmed all over the Midwest of the US as well. So we really know that none of those traits work on Western mean cutworm, and that Viptera is the only one that still does. However, there's a caveat here in that we've looked at the susceptibility of really young Western bean cutworm larvae to VIP versus the older larvae. And we can document that it takes a couple hundred times more protein to kill the LC50, to kill a third or fifth instar Western bean cutworm larva compared to a first. So this is why we're a little bit concerned about the potential for resistance to develop to VIP3A, especially in a refuge in a bag scenario where an insect may develop on a refuge plant and get to be a third or fourth or fifth instar and then move to a VIP plant. They will be able to tolerate that VIP protein. So we are a little bit concerned about that because Western bean cutworm do move around quite a bit in the field. So we're recommending that we don't solely rely on VIP hybrids to control Western bean cutworm going forward. We think that's a bad strategy for resistance management. So we're recommending that growers alternate between using VIP some years and maybe just using a non-BT hybrid or one of the other BTs that aren't controlling Western bean cutworm, and scout and spray. So you're going to be scouting for egg masses in late July, early August, and if you hit a 5% threshold, then you would use an insecticide application. We have some years, and it varies by region, where Western bean cutworm is not an issue in Ontario, and in other years it is. So that we still don't think it's a pest that's always there. So this is maybe a good strategy that will help us preserve these products for long-term use. For corn earworm, it's kind of a similar story. I put this in a little different way. So we know from the beginning that Cry1F never controlled corn earworm. In the early days, Cry1AB, Cry1A.105, and Cry2AB2 did control corn earworm, and VIP3A does control them as well. Unfortunately, though, we're seeing this really shifting in recent years. So the sentinel plots that Sonny mentioned in his video, where we're monitoring resistance for corn earworm, they were originally started by a researcher in Maryland who was doing these since 1996. And in these little graphs here, the blue lines are the Cry1AB sweet corn hybrids, and the yellow lines are the BT hybrids. Now, this is percentage of damaged corn ears in the top left graph. We'll just look at that one quickly, and you can see back in 96 how there was almost no damage on the Cry1AB hybrids, but by 2016 there was quite a bit. We were talking 65–70% of the plants with injury. So this is also showing that the instars or the size and development of the larva has increased over time on Cry1AB, so we know we've got resistance happening there. And this is the same story with the Cry1A.105 and 2AB2 traits in the Smart Stacks hybrids. So unfortunately, we now say that the VIP hybrid is the only, or the VIP trait is the only one that will control corn earworm as well as Western bean cutworm. So those two insects are pretty similar. And lastly, I'm just going to talk about European corn borer for a minute. Originally, this was the main target pest for BT. And all of these cry proteins controlled European corn borer. Interestingly, the VIP trait has never controlled European corn borer, so that's just something to keep in mind. And since this graph on the right shows that these are corn borer populations year over year in some of the corn states, and the red line shows us when BT corn was first introduced, which was primarily Cry1AB corn. And you can see that the corn borer population since that time really crashed in some of those states. And the gray bars in the background are the adoption of BT corn since then. So we know that the BT traits really, these are all high dose traits against European corn borer. They kill 99.9% of the insects that feed on it, and it really knocked back our populations. However, in 2018, we were notified of a bit of an anomaly in Nova Scotia, where around Truro, there were a number of fields discovered that were Cry1F hybrids, and there was anywhere from 30 to 70% of the plants with European corn borer injury. And there's some photos from those fields. So what we did was we went out to collect some of those populations. We bring them back to our lab. We rear them up on diet and so on, and then we do these bioassay tests where we expose them to different concentrations of the proteins, Cry1F in this case, to determine the LC50 of the populations. And so here are the results from those studies. Basically, if you just want to look at the lines on the graph, this is mortality on the y-axis, and we had 100% mortality of 2 Ontario populations, as we would expect, with a concentration of, by 30 nanograms per centimeter squared of Cry1F. And so our LC50 for those populations was about 6.5. However, with the 4 sites around Truro, we couldn't kill them. We had more than 80% survival of those populations with up to 200 nanograms of Cry1F. And we also had a collection here from Kentville over in the Annapolis Valley, hoping that it would be susceptible as it's kind of downwind from or upwind from the resistant populations, but unfortunately it showed up as resistant as well. We've since tested these on plant tissues as well, and we get complete survival of the 4 populations from the Truro area. The Kentville population does still die on the tissues, so they're maybe not completely resistant at this point, but they're on their way, unfortunately. And so, there's a number of questions about why on earth did this happen in Nova Scotia when we've been monitoring corn borers since the 90s around most of North America, and we've never had any resistance problems like that develop. However, we don't know if there's a bit of an island effect there where your populations have been isolated and there's been heavy selection pressure placed on them. We know that, we found out as this all came to light that there were a lot of single BT trait hybrids being sold in some of these smaller short season markets, such as the East Coast, such as in Manitoba, and now we know this has been the case in some of the US as well. Unfortunately, and in the rest of North America, as Sonny mentioned, most of the hybrids you buy have a pyramid, so we have less chance of resistance happening. But in this case, we had still a lot of singles being used, so that probably played a big role here. Perhaps there was not enough refuge implemented. We don't really know about that. And then, it was kind of under the radar. I think we've not had good enough communication with our colleagues in the East Coast in the past, and the resistance monitoring for European corn borer was more targeting the large market areas for corn production. And we can see now that this is a mistake, and we need to look at some of the more unique scenarios as well. So that's what has happened there. And what we're recommending is that those single trait hybrids are no longer sold, and that they have been phased out since 2019 in the East Coast, and pyramids have replaced them. Hopefully we can get more pyramided products out there sooner than later. If there are resistance issues in a field, the corn stalks should be chopped and buried in the fall to reduce the survival of potential resistant populations. And then we're going to really increase our work out in the East Coast, studying European corn borer populations and their susceptibility to the different proteins, and also just their basic biology, because we don't know a lot about what other plants they're maybe using as hosts and so on. So in 2019, we've got a lot of collections out there, which we'll be testing in our lab, and we will keep you posted on things. So just to summarize, there's the the breakdown of all of those traits again. And what I want to point out here is this column on the right, which shows that in terms of future traits coming to control these pests, there's nothing close to the market. It's going to be 10 plus years before we have anything. So we need to preserve what we have. So we need to do a better job with this resistance management program. And I just put the corn rootworm down at the bottom there as well. There are a number of traits, and there are a lot of resistance problems happening with that in the US right now. And unfortunately in Ontario, we're starting to find this as well. So where you have a lot of continuous corn production, if they're using these traits year over year, they're at high risk for resistance. So again, it comes back to rotating your management strategies. Okay, and I'll just leave it there. We can take questions later, I guess.
Great. Thank you so much, Jocelyn. That was great information. Definitely, definitely one of those subjects that we're going to follow closely as things develop. All right. So now we're going to have a bit of a question period. If you want to unshare your screen there, we'll get our speakers up. Hopefully you can see some of our lovely faces. And wonderful. Welcome. Welcome, welcome. I think I will kick things off with a question for Suzie. So I'm wondering, Suzie, if you can tell me a little bit more about where the insects are feeding on the cucurbit plants and kind of some of your theory behind that.
Okay, we notice with, we'll start with cucumber beetle. That's the first one to show up in the season. And you will readily find it, I mean, as the picture showed beautifully, that they feed, you know, they'll go right after the blossoms. They'll feed on the blossoms, they'll go after the leaves. And then once the eggs are laid, and they're laid at the soil surface, the larvae then burrow into and will feed on the roots. So even if you don't get the bacterial wilt happening, which can happen, you may end up with your leaves looking wilty simply because there's holes in the root system. So they're having a harder time getting the water that they need to maintain their turgor. So that's with your cucumber beetle. With the squash bug, I mean, I know that it talks a lot about the adults feeding not only on the leaves, but also on the fruit or the squash itself. And I would say with us, with what we've been seeing for the butternut squash in particular, that the way they're partitioning their use of that resource is they're treating the leaves kind of like their kitchen, and then they're heading over to this fruit and they're just hanging out there and basking away in the sun, and it's more like their living room. Because despite seeing dozens of squash bugs sitting on a butternut squash, we're not seeing any damage from them. Now, I think it might be a little bit different over in buttercup, but it's hard to tease out who's doing the damage on that fruit, because cucumber beetle is there definitely first, and they've had a longer time period. And this year, where they had 2 generations, they had 2 cracks at the fruit. So it's hard to know if we were having the damage we're seeing is kind of compounded by the 2 species, or if it's largely just cucumber beetle.
Mm-hmm. Great, thank you. We do have a question, what varieties of squash were planted? So we had 1 butternut and 1 buttercup.
Yeah, so it was it was the Burgess.
Burgess buttercup. Yeah.
Burgess buttercup, and then it would have been the Waltham butternut.
Wonderful. Thank you. All right, Francisco, I'm going to toss it over to you.
Okay, thank you, Kate. Yes, I have some questions for Dr. Deb Moreau. For example, how is propagate a phylloxera through the vineyard?
Once it's in your vineyard, so it can come in on infected plant material. So recent expansion efforts in Nova Scotia in vineyards, that definitely posed a risk. If you're bringing in plant material from sources that maybe didn't capture that, then you're now introducing them there. They can also come in from pre-existing, old blocks that are acting as a reservoir. But once there, typically the crawler, so certainly the root stage will can actually crawl out of the soil. They take advantage late summer when the soil sort of cracks under the vines, and they can move up to the top. They can walk along the soil surface. They can climb the vines, and they can also be pushed around considerable distances by wind. And then, of course, human efforts can further move that in and around the block. So tilling can do that. Often if infestations are really high, there are lots of sightings in the literature that suggest that individuals can be picked up in the dirt on your boots, equipment, picking buckets, those types of things, and then move from block to block. So in Nova Scotia, where a lot of vineyards that are more mechanized and they use the knife hose and cultivators, that definitely poses a risk, for sure.
So it sounds like biosecurity is an important factor there with spreading it around.
Yeah, phylloxera is one of those, I would argue, and possibly folks on this call would agree that, many economic pests are very, I would argue it's an ideal to eradicate. So phylloxera is something that once it's here, it's here, it's a resident. Mm-hmm. So sanitization is huge. So, just kind of off top of mind, one recommendation is always, if you have a block that has a severe infestation, then when you're planning your work within the vineyard, save that block to the end. Don't cultivate that first and then move into another block. And that's just a suggestion.
Okay, wonderful. All right, we have a couple more questions coming in now. Do systemic chemicals like Movento kill aphids in the root as well? Toss that out to whoever wants to answer it.
Yeah, they certainly, Movento is a really great product for definitely phylloxera, and certainly, whether they're feeding on the roots or they're in galls on the leaves and feeding on the tissue, they'll certainly pick it up. It is not an option for organic growers, and a lot of vineyards in Canada, actually, in the Canadian wine grape growing regions, are moving towards organic. Mm-hmm. So that's not an option. And at this point in time, although it's effective, you would only get 2 kicks of the can in a season with that product. You have to wait 30 days in between sprays. And certainly in my readings, it is suggested that once you start using Movento, you kind of have to use that for a number of years to kind of get good suppression. And again, it will suppress, it will knock down populations, but it will not eradicate from the farm or from the vineyard. I can't speak to the other aphids.
Great, thank you. Suzie, another one for you. How mobile are squash bugs from year to year, and how much distance is needed between plantings to help prevent movement from last year's planting into this year's?
Good questions. The interesting thing, when we set that trial up at, so it was set up at the Kentville Research and Development Center, and it's right next to the vineyard. So if you're familiar with where the vineyard is at the station, we're just in the plot to the west of it. The only cucurbits that we are aware of that were grown on our acreage were a pumpkin trial that we had going on, and it was on the other side of the property, on the eastern side, just above where the picnic grounds are. So we had basically the two opposites. So when we set the trial up, there was concern that we wouldn't necessarily, are we going to see them? Are they going to show up? One of my technicians said, well, I've got loads of them at home, so we could just import them if we don't get them, which we didn't have to do. They easily covered that distance. So I would say you're looking at kilometers of distance in order to get away from these populations, if you know that they're nearby. And the other thing is, when if you're up next to or near any urban home gardeners, there's always potential for those to have a bit of a sink where they can then move into what you've got going.
Mm-hmm. Yes, tricky little things. Great. I have a question for Jocelyn. What other management strategies other than the pyramid traits are going to be most important for growers in maintaining the longevity of the current traits?
Well, you're talking about, well, I guess we could think about it pest by pest, or we could try to generalize this. And I think, you know, the same thing with insecticide resistance, you know, we want to think about rotation, and that's your main strategy. And we've kind of gotten ourselves into a bit of a problem here with the pyramids and the cross licensing of the pyramids, in that all of the companies now have cross licensed the traits to each other. So rotating is becoming more of a challenge. It's a bit of a conundrum, actually. So I guess in this case, you know, you really need to sit down with your agronomist or your seed dealer and kind of think about a long-term strategy, looking at crop rotation being the first choice, I would say, especially if you're talking corn rootworm. You want to keep crop rotation in there as much as you can, because if you're going to rely on the same traits for, a single trait on corn rootworm for 3 consecutive years will have resistance. So when we pyramid, we're gaining maybe a couple more years, but I think that's not a great long-term strategy. So you got to consider the crop rotation as the main thing. And then you maybe consider not using BT sometimes, and just using thresholds and insecticides as control methods. Yeah.
Great. Yeah, I think it's definitely a big picture view on things from here on out.
It is, and with the other pests, which are all moths as adults, they're highly mobile, right? So they're going to move around a lot. So crop rotation isn't always going to be a good solution for you. So you got to think about traits and other things.
Wonderful. Okay, Francisco, do you have any other burning questions?
Yeah, I have another question for Deb, because she was saying, or thanks to the previous question about how to manage phylloxera in a conventional vineyard, but which options, or how is the best way for an organic producer, especially if he has a Vitis vinifera variety?
So ultimately, the best plan is to have your vinifera grafted on resistant rootstock. That said, you know, resistance in rootstocks in terms of phylloxera feeding is kind of a mixed bag. So it's a moving target. Some rootstocks are better than others. It's really a degree of susceptibility to the feeding. So ultimately, though, putting your vinifera on rootstock that offers you some level of resistance is optimal. If that's not the case, then it really comes down to, and this really applies to conventional also, it really comes down to monitoring. So if you've established that your block has an infestation, then really the next step is to do an annual survey every year. I would recommend probably late summer, early fall. You could probably push it to after harvest. I know folks are swamped at harvest, and kind of walk through and identify and look for the above ground symptoms. So things like over time, you can see in some varieties a decline in pruning weights and yields by vine. So if you start to see symptoms that suggest less vigor and impact on the overall vine health or performance, it may be something worth noting and kind of monitoring. But then looking for where you're seeing that sort of poor vine health, and then revisiting those vines every year and sort of tracking to see. That's a really kind of loose way to look at kind of that rate of spread and movement through the vineyard. So Phylloxera don't necessarily go up and down the rows perfectly. If you were to fly over a block that had a severe infestation, see is little patches that almost look like a contact lens, like a little lens shape. So it'll be a series of vines. I would not focus on the vines that look like they're near death, because the phylloxera will, when the vine starts to get quite severely compromised, they'll go to healthier vines. And sort of just track those little pockets over time, and usually there's a directionality to the spread through your vineyard. And then I think with that information, then you can make a decision in terms of whether it's worth your while to pull out the block. And I would not recommend roguing, pulling out individual vines. In all likelihood, you're going to leave bits and pieces of the root systems and phylloxera, and they're just going to move to the next healthy vine. So if down the road, economically speaking, it's important to, that the vines have clearly been compromised and you're risking losing the whole block, then you would want to know kind of the level of the damage. So it's really a lot of monitoring, and then there's a long list of sanitize, to Caitlin's earlier point, things you can do just in terms of your day-to-day actions within the vineyard to minimize moving it from one block to another.
Mm-hmm. Great.
It's a tricky, it's a very fast, it's a very...
Yeah. We have a comment here about predating insects for phylloxera. Do you have any comment on that?
I didn't catch your statement, Caitlin.
Predatory insects for phylloxera.
So it's a relative of aphids and kind of, that great mealybugs, scale insects, that group of insects. There are generalists out there that are good, that certainly if they intercepted one of them, I think green lacewings and certainly ladybird beetles, things like that. The issue with phylloxera is that at any one time, a proportion of the population will be in the ground, in the roots, and a portion of that same population will be in the leaves, and they are very well protected within the galls. So the vulnerable stages, which are really the crawlers when they move out of the soil, and kind of those that are moving from the leaf galls down into the soil, that's really the vulnerable stage. So with general predators, yes, absolutely, there are all kinds of beneficials out there that if they intercepted, they would gobble them up. But there are also, that one is kind of a hit and miss in terms of, sustained control or suppression. But there are some handy ways, certainly that I've discussed with the industry before, double-sided sticky tape that you use to put carpet down, wrapped around the trunk, kind of where the cordons branch off, is a really effective way. The crawlers get stuck on it. So there are a little bit more time, it takes a little more time to do it, but if you're not monitoring and having to check it, it's really just an issue of wrapping the tape around and then removing it every couple of weeks and refreshing it. So there are some ways to kind of catch that vulnerable stage, but predators, there would be a lot of other competing factors to allow them to actually suppress the populations.
Right. Okay, wonderful. Well, we had another question about crop rotation for managing the corn's pests, which Jocelyn has answered there in the comments, if anybody wants to read that there. But other than that, I think that wraps up our question period. We're kind of right where we wanted to be time-wise. Does anybody else have any burning questions or last comments before we wrap up? No? All good? Well, thank you all very much for tuning in and joining us on our virtual field day. And a big thank you to our 3 guests today. It was wonderful to have your expertise. And also a big thank you to Rachel, who is our marketing person, and she was both our videographer and our background producer technical support during all of this. So thank you very much, and we hope that lots of people can join us the rest of the week. Just as a reminder, tomorrow we're doing production systems, and on Thursday we are doing weed pests in various horticulture crops. So thank you all very much, and hope to see you soon. Bye.